Welcome to On-Demand Learning for FlowCam 8000. In this video, we will demonstrate the proper way to install and remove the field of view FOV flow cell. We will first describe how to install the flow cell, and then we will describe how to remove the flow cell. In your hardware and accessories case, you will find your magnifying objective lenses, microfluidic tubings, field of view FOV flow cells, and pump syringes. Your FlowCam is calibrated at each magnification with defined accessory pairings. You can find this chart in the FlowCam 8000 series configuration guide, a technical note in your desktop resource folder. In this demonstration, we will be using the 10x magnification lens with the FOV 100 flow cell and the 1 ml syringe. Remove the flow cell from the protective case. The upper steel fitting is marked with a color stripe. Confirm the flow cell size by reading the FOV inner channel dimensions etched on the front face of the flow cell near the upper steel fitting. To install the FOV flow cell, you will need the flow cell, the upper microfluidic tubing, and the longer lower microfluidic tubing. Pick up the flow cell and hold it firmly by the upper steel fitting. The upper microfluidic tubing is the shorter of the two tubings. Grab the upper microfluidic tubing and thread the click and seal fitting into the upper steel fitting of the flow cell. Once firmly screwed in, give an extra twist to the fitting until you hear and feel a click. This ensures that the correct torque is reached and the tubing is properly sealed. In the same way, connect the lower microfluidic tubing to the lower end of the flow cell. Hold the flow cell by the lower steel fitting and screw in the click and seal fitting. Once firmly screwed in, give an extra twist until you hear and feel a click. Remember to only hold the lower steel fitting and the click and seal fitting when tightening the connection to the lower tubing. Holding the flow cell elsewhere can introduce excessive torque and cause the flow cell, which is made out of glass, to break. To prepare the flow cell chamber, unscrew the red-tipped thumb screws to release the flow cell retainer. Remove the flow cell retainer and inspect the chamber. Make sure the thumb screw tips are completely clear of the chamber to prevent any interference with the flow cell during installation. Now that the flow cell chamber is prepared, you may install the flow cell. With the flow cell color stripe and etching facing you, hold the upper microfluidic tubing taut and securely connect the click and seal fitting to the fitting below the sample port. Keep the flow cell out of the flow cell chamber as you proceed to connecting the lower microfluidic tubing to the pump manifold. With both the upper and lower tubings securely connected, press the upper tubing into the fluid position sensor using your finger or thumb. The leading and trailing edge of the fluid sample are automatically identified by this sensor, so it is important to have the tubing positioned snugly into this part of the instrument. The flow cell is now ready to be gently placed all the way to the back left side of the flow cell chamber, leaving room on the right for the flow cell retainer to slide in. Use one hand to keep the flow cell flat, straight, and at the back left side of the chamber as you slide the retainer in along the right side unobstructed. If the retainer does not slide in smoothly, remove it and reposition the flow cell before trying again. Do not force the retainer into place. Complete the installation by tightening the thumb screws to finger tightness. Do not over-tighten. You can check the flow cell alignment with the camera's acceptable region by opening the camera view. In this live image of the flow cell, you can see where the flow cell inner channel, the black lines, are relative to the camera's acceptable region, marked by the red line border. The camera only sees what is inside the red border, so you want to make sure the flow cell is properly aligned. To adjust the flow cell alignment manually, open the objective shroud and find the mirror knob. Turn the mirror knob very slightly as you observe the camera view display. Continue adjusting until the black lines marked by the flow cell inner channel edges are just outside the red lines of the camera's view. You can also fine-tune the flow cell position using a feature within a visual spreadsheet. In Camera View, go to Setup and select Capture. In the Acceptable Region panel of the Capture Setup dialog, change the left and right fields and click Apply to see the effects of the changes in the camera view. You may have to apply a couple of changes before the ideal positioning is achieved. Once the flow cell is appropriately positioned within the camera's acceptable region, you can close the Capture Setup dialog box and close the camera view. The installation of the flow cell is now complete. To remove the field of view FOV flow cell, begin by removing the flow cell retainer, then working bottom to top. Loosen the red thumb screws until the screw ends are clear from the flow cell chamber, and carefully remove the flow cell retainer. Make sure the thumb screws are fully clear of the flow cell chamber to prevent flow cell damage. Carefully draw the flow cell out of the chamber and disconnect the lower microfluidic tubing from the pump manifold. With the flow cell drawn out of the flow cell chamber, release the upper tubing from the fluid position sensor. Holding the upper tubing at the top of the flow cell with one hand, use the other hand to unscrew the upper tubing from the sample port. Removing the field of view FOV flow cell is now complete. Detach the upper and lower microfluidic tubing from the flow cell, holding the upper and lower steel fittings respectively. Return the flow cell to its protective case and place it in the hardware and accessories case for safe storage.